Yeah, so today I wanted to present our work on the dynamics of engram cells and systems consolidation. So the idea here is based on this experimental finding that engram cells in medial prefrontal cortex and in hippocampus, they behave differently. So in medial prefrontal cortex after training, engram cells in medial prefrontal cortex, they are in this silent state, meaning that they can't be recalled at a recent time point, but then over time, they become active and then now they can be recalled at a remote time point. And this process of going from silent to active is termed maturation. And then it is accompanied by this increase in dendritic spine density. But then for hippocampus engram cells they're encoded already in an active state, meaning that they can be recalled at a recent time point. But then over time, they become silent and they can no longer be recalled in a remote time point. So this process of going from active to silent is termed dematuration, and it is followed by this decrease in dendritic spine density. So our goal in this project is to investigate the mechanisms behind these engram cell dynamics. So we started with the simplest possible model, which only includes hippocampus and cortex. And then the idea here is to replicate engram cell dynamics in both of these regions, and also to capture the fact that the output of hippocampal engram cells is essential for the maturation that happens in cortex, as demonstrated in the experiments. So we start the simulation with a training phase where we present 4 distinct stimuli at random. Then we go to a consolidation phase where we don't present any stimulus, the network evolves spontaneously. And then finally, we go to a testing phase where we present partial cues. Then we measure the ability to recall the engram cells in both of the regions. So at this point, I wanted to point out that even though the synapses in the network are plastic, the synaptic plasticity rate is region specific. So meaning that the learning rate in hippocampus is higher than the learning rate in cortex. And also the synaptic consolidation time constant in hippocampus is also longer than the one in cortex. So here we can look at the degree of coupling between hippocampus and cortex at the end of the training phase. And then what we can see is that by looking at the mean weight strength between the engram cells that encode each of the stimuli between these two regions, that they have very strong coupling, such that after consolidation, when we do testing, we can see that here in blue, hippocampus has a very high recall immediately after training, and then has a very low recall after consolidation has taken place. So we have dematuration in hippocampus. But then now when we look at cortex, it already has a very high recall even right after training. Then after a small decrease, it goes back up. So this is not a profile of maturation as it should be, going from silent to active. And then here, if we block the output of hippocampal engram cells to cortex in the consolidation phase, we can see that we can recover a very high recall in cortex throughout the entire consolidation period. So this configuration here cannot display engram cell maturation in cortex as it should, based on the experimental data. So what we did was we fixed the connections from hippocampus to cortex. Now they're not plastic anymore. We go through the same simulation protocol, and then now, as expected, we do not have coupling between these two regions anymore, between hippocampus and cortex, since these synapses now are fixed. And then when we do the testing with the intact network, now we do have maturation of cortical engram cells, because initially here in orange, they're silent, and then later after consolidation, they're active. However, when we block the output of hippocampal engram cells to cortex, we still have the same profile of maturation, meaning that hippocampal engram cells are inconsequential for the dynamics in cortex. And this is contradictory to the experimental data. So in conclusion here, using this simple architecture with only hippocampus and cortex, we cannot capture this effect of hippocampus being critical for systems consolidation in cortex. So a solution to this is to include an intermediary, the medial dorsal thalamus specifically. And now we have this multisynaptic pathway that goes from hippocampus to thalamus to cortex. And then by using this expanded network, we can replicate dematuration in hippocampus, maturation in cortex, just like before. But then now the output of hippocampal engram cells is critical, is essential for the dynamics of engram cells in cortex, matching the experimental findings. So in this configuration here, we use these 4 set of stimuli, these 4 horizontal bars. And then after training, we go through consolidation, no stimulus. Then we go to testing, where we present only the middle 50%. And then in the consolidation phase with the intact network, here in orange for cortex, we can see that we have, again, maturation going from silent to active, and dematuration going from active to silent in hippocampus here in blue. And then we can see here that the model also predicts that engram cells in thalamus are going to be active at recent and remote time points. Now we can look at what happens to the engram cells during the consolidation period. So here in the last half an hour of the 24 hours of simulation in the consolidation phase, we can see here that in cortex, thalamus, and hippocampus, we have these coupled reactivations of engram cells. And then now we can do repeat the simulation, but then now block the output of hippocampal engram cells to thalamus. And then what we can see is that we no longer have proper maturation of cortical engrams. Instead, here in our orange, you're going to have this small increase in recall in cortex, and then after that, it quickly declines, and essentially, you cannot sustain proper maturation of engram cells but by excluding the output of hippocampal engrams. So essentially what this is saying is that you need to have hippocampal engram cells active during consolidation for proper engram maturation. And then here, when we look at the output, the activity of the engram cells in the consolidation period, again, in cortex, thalamus, and hippocampus, we can see that blocking the output of hippocampal engram cells disrupts that coupling that we had before in the control network. So the idea here is that the model suggests that engram cells in hippocampus are essential for dynamics of engram cells in cortex because it is necessary for coupling reactivations across these regions. So now we can repeat again our simulation with this network, but now we block the output of thalamic engram cells to cortex. And then we can see here that we're going to have a very similar effect, but in a slower fashion. So we have this increase in cortical recall followed by a decrease, but even though we have this initial increase, eventually, gradually it goes down. So essentially, we also cannot have, do not have in this case, a proper engram cell maturation when we block the output of thalamic engram cells. So these thalamic engram cells are also essential for maturation in cortex. And we can see that the way that they support maturation in cortex is also by coupling reactivations across the regions, because when we block the output of thalamic engram cells, we no longer have coupled reactivations between cortex and thalamus. And this is the reason why we have this decline in accuracy in cortex that we see here. So medial dorsal thalamus engram cells are essential for cortical engram dynamics, just like hippocampal ones. And now finally, what we wanted to study is the role of hippocampus in recent and remote recall. So what we found here is that the degree of synaptic coupling between medial dorsal thalamus and medial prefrontal cortex can predict the dynamics of engram cells in medial prefrontal cortex. And as a result, it can also tell what kind of retrograde amnesia pattern is induced by hippocampal ablation. So let's walk through this step by step. First, we have the training phase, just like before, and the consolidation. Now in the testing phase, we do it in two ways. One with the intact network, and then in another case, we remove hippocampus just in the testing phase to see the effect of that on recall. So at the end of training, we can measure the coupling from thalamus to cortex, and then we can see here that it's strong, relatively strong, and then after 24 hours of consolidation, it gets even stronger. So this gives us this profile of recall that we saw before, where here in orange for cortex, again, we have maturation going from silent to active cortical engram cells. However, when we remove hippocampus from the network in the testing phase, we can no longer recall the memory at a recent time point because recall in cortex is very low. But then you can recall the memory at a remote time point because by then we already have active engram cells in cortex. So this pattern is here configures a graded retrograde amnesia. But then we can control the degree of coupling from thalamus to cortex by controlling the synaptic plasticity rate between these regions. So by modulating this plasticity rate, we can then have very weak coupling from thalamus to cortex at the end of training, which does not change after 24 hours of consolidation. It is virtually non-existing, the coupling between thalamus and cortex in this case. And this leads to a very different profile of recall for cortical engram cells. You can see here that they have a low recall in recent and remote time points. So you do not have that maturation that we had before here. And then when we do the testing with hippocampal ablation, we cannot recall the memory neither in recent nor in remote time points. And therefore, we have a flat retrograde amnesia pattern. Finally, we can modulate the coupling from thalamus to cortex to be stronger by controlling, again, the synaptic plasticity rate between them, such that at the end of training, you have very strong coupling, even stronger than we had before. And after 24 hours of consolidation, this coupling only gets stronger. So now we have a different profile of cortical engram cell recall here in orange. We can recall memories in cortex in recent and remote time points. And then now when we do hippocampal ablation, we can still do that, and therefore we don't have retrograde amnesia anymore. So basically we have an absent retrograde amnesia curve. So all this is to say that thalamocortical coupling, the degree of synaptic coupling between medial dorsal thalamus and medial prefrontal cortex, at the end of encoding is driving the dynamics of engram cells in cortex, and therefore the retrograde amnesia pattern that is induced by hippocampal lesion. And then finally, to conclude, the model was able to reproduce some key experimental findings in the literature of engram cells, specifically engram cell maturation in cortex and dematuration in hippocampus. The fact that in hippocampal engram cells and the coupled reactivations are necessary for proper systems consolidation. And we also make a number of testable predictions, and some of which here are engram cells in medial dorsal thalamus are active in recent and remote recall. The fact that thalamic engram cells are necessary for proper engram cell maturation in cortex. The fact that engram cells in hippocampus and thalamus are necessary for coupling reactivations across the hippocampus thalamus medial prefrontal cortex pathway. And finally, that thalamocortical coupling is predictive of the type of retrograde amnesia that is induced by hippocampal lesion. Thank you. If you have any questions.
You're muted. You should ask you on mute.
Okay, I guess now I can. I thought it was my problem. I was not hearing anything. So.
I'm sorry, I'm muted. Thanks very much for your talk. That was excellent. I'm not taking questions from Q&A. We have a question from Shruti. Can you tell us a little more about the networks you have used and designated as hippocampus, cortex, and thalamus?
Sure. So if we look at this configuration here, which is the one that has all the three of them, you guys can still see my screen, right? By the way.
Yes.
Okay. Yeah. So so the way that we tell that a cell is an engram cell is that at the end of the training phase, we take the last 5 minutes of training, and then we calculate the average firing rate that is evoked by the stimuli. And then if this average firing rate over the last 5 minutes is above a certain threshold, in our case 10 hertz, then we say that this cell encodes that specific stimulus. So any given cell can be an engram cell after a certain period of training, and then the same cell can encode multiple stimuli as well. And then the difference between the network in hippocampus, thalamus, and cortex is purely in terms of synaptic plasticity rate and in terms of the afferent and afferent connections, as you can see here in the schematic. So in terms of what projects to what, and then and also in terms of the learning rate and the other parameters that control the synaptic plasticity rate in each region. That I hope that clarifies it a little bit more.
Cool. Thanks very much. Douglas, can you also click Q&A to see the questions? And Shruti also said one more thing more specifically, but I think you already answered that question. So we go for next.
Yeah, I can see it here.
The question from Diogo Pat, yes.
Yeah, so have you considered to close the cortex thalamus CA1 loop via nucleus reunions? No, I have not. That's a very interesting idea. We were kind of like starting with the simplest possible model, and then we added things that that made the minimal circuit that could reproduce these engram cell dynamics. But certainly there's a lot of work to do, and it include other thalamic nuclei and also other cortical areas. That's something that we're considering for future work.
Okay, cool. Thanks very much. That was really great. But we have a very tight schedule, so